Hello everyone. So in this video we will be covering about the epithelial tissues in detail. Like in the previous video, I have told you about the basic definition of tissues and how many types of tissues are there in our body. So in this video we will be covering with the first type of animal tissue, which is epithelial tissue. So I have written all the types of epithelial tissue that we will be covering. So one by one we will cover all this. So simple epithelial tissue, the types you can see: squamous, cuboidal, columnar, ciliated, glandular, and pseudostratified. And this division is on the basis of their structure as well as their function. Now, if I talk about epithelial tissue, simple, I have told you, with a compound. Compound means they will have, uh, different layers, and that is having more than one layers. So first is stratified and second is transitional. Stratified again is divided into squamous, cuboidal, and columnar. The squamous is divided into keratinized and non-keratinized. And transitional, this particular epithelium is stretchable. We can stretch it to a bit. Okay, so from the squamous simple epithelial tissue, we will be starting. So let's see what is the exact diagram of simple squamous epithelial tissue. The most important point is that this is a flat layer. They form a flat layer. This is thin. Obviously, it is made up of single cells, so this will be thin layer made up of single layer, and that's why the name simple is given, because it is made up of a single layer. So we can make a idea why single layer is present. Because of the single layer, diffusion and filtration can take place. So the most important function, because this point is coming from this particular point only, it is a single layer because some important function has to be performed, like filtration, and exchange of gases. All right. Now, talking about the exact structure of the simple epithelial tissue, the structure is like this. These cells will have a wavy margin. As you can see in the diagram I have drawn, nuclei will be in the center. So this is irregular boundaries. So a particular term is given for the irregular boundaries. We call it as tessellated epithelium. This is also known as tessellated epithelium. Okay. Now, functions, filtration, exchange of gases. Now, from this point, we can make an idea from where you will find this type of epithelium. You will find it location alveoli, where the gaseous exchange take place. Then, in the lining of blood vessels, and lymph vessels. From where the exchange of material between the blood and the fluid present around the tissue, it take place. And in the Bowman capsule, of kidney. So these are the location where you will find this type of thin epithelium, squamous epithelium, where the exchange of gases take place in the alveoli, from where the materials will exchange between the blood and the fluid of the tissues, and the Bowman capsule, from where the filtration of the blood it will take place and formation of urine will take place in the kidney. Okay, now after squamous epithelium, let's talk about simple cuboidal epithelial tissue. As the name suggests, they will be of a cuboid shape. Now, let me draw the shape. Suppose it's the basement membrane. In every epithelial tissue, you will find the basement membrane. Now, the cells are of cuboid shape. Some will be simple. Some may have some finger-like projections, like this, and some may have hair-like projection, which is known as cilia. Nuclei is round in the center. Now, these type of cells, it's the simple cuboidal because nothing is present on the top, as you can see, simple cuboidal. And this particular type of cuboidal, we call it as a brush border epithelial tissue. Brush border epithelial tissue. And this is known as ciliated cuboidal. Okay, now simple cuboidal epithelial tissue you will find in certain parts. For example, you will find it in the small ducts of gland, thyroid follicles, germinal epithelium. This is a very important point. Germinal epithelium. It means testes and ovaries. And again, important thing, you will find it in the ascending limb of LOH, in the ascending limb of loop of Henle. One more important thing that I would like to add, in the previous squamous epithelial tissue, you will find that in the descending limb of LOH as well. That was thin. This is a bit thick. So you will find this simple cuboidal. So LOH is having one thin and one thick. In the thin, you will find a squamous. In the thick one, you will find this particular simple cuboidal epithelial tissue. Brush border epithelial tissue, because the main function is absorption of this brush border. As you can see, these are known as the finger-like projections are known as microvilli. So microvilli are found in the area where absorption has to take place. So you will find it in the nephron. The maximum absorption takes place in the PCT, proximal convoluted tubule. This is a very important point. Now, ciliated cuboidal tissue, which we also call as kinociliated. The cilia, no? Kinociliated, because cilia they will move, so that provide kinetic energy. That's why this word kinociliated is being written. So you will find these in terminal bronchioles. Okay, so from this every piece of knowledge, you will find that the basic function of these tissue is absorption. Okay. and secretion as well. As you can see, they are found in the germinal epithelium. So they are the gland they form secretions. So this is the main function of simple cuboidal epithelial tissue. Now we will be covering about the simple columnar epithelial tissue. Now from the name, as you can see, again I will draw this basement membrane and some elongated cells. I will draw because of the name suggests columnar. They are column-like. They are elongated. Again, you will find these finger-like projections projected from the brush border, and you will find ciliated one. And there are also cells which will have cilia, but the cilia will not move. It will be static at one place. So this is cilia there. It can move, and if the simple cilia cannot move. Okay, so this is a simple columnar. This is brush border columnar. This is kinociliated, or you can say simply ciliated columnar, and this is stenociliated. Cilia is there, but cilia will not move. Simple columnar epithelial tissue, you will find the glands of the GI tract, glands of gastrointestinal tract. After the esophagus, because absorption it occurs, so where the absorption will take place, you will find these simple columnar epithelial tissues. Brush border. You will find the brush border, but the absorption will take place after esophagus. Okay. So in the glands, you will find your simple columnar epithelial tissue. Brush border you will find in the GI tract. Okay, you have already studied in your digestion chapter that these particular type of brush border epithelium having microvilli, you will find it in small intestine. But other parts also you will find. This is microvilli again. Now, ciliated columnar, you will find cilia moving. You will find it in fallopian tube because they have to push the egg to the, into the. They have to push the egg for the fertilization with the sperm. Okay, so this is the first thing where you will find the ciliated one. Again, another is in the upper respiratory tract as well. In order to move the dust, okay, in order to protect you from the dust particles. Stenociliated, there are some examples of it, like in vas deferens and in epididymis. You will find these stenociliated type of columnar epithelial tissues. Okay, so from all this, again, we will conclude what is the function of the main function of this particular type of epithelial tissue is absorption. Okay, so we have covered about important three types of epithelial tissue: squamous, cuboidal, and columnar. They differ in their shape and in their function also. And with the diagram, you can correlate what will be the location, and from the location you can also correlate what will be the function of this type of epithelial tissue. Now, there are two types of epithelial tissue still left, which is pseudostratified epithelial tissue and glandular. Because ciliated, we have already covered with these type of epithelial tissue. The rest we will be covering in the subsequent video. So please, you can see the next video for this. For any doubts in this particular video, you can write it on the comment section. Thank you so much.